Neuroscience and beyond. Know more, get inspired. Welcome to the Neuroscience and Beyond podcast. My name is William Georgiev and I'm a PhD student at the University Medical Center in Göttingen, Germany. Together with my PhD colleagues Sabina and Christina, we desire to bring science, and especially neuroscience, closer to the general public. In the following episodes, we will talk to various scientists about their inspiration to do science, their research topics, and their opinion on current scientific problems. Today, our guest is Ralf Heinrich. He is a professor at the University of Göttingen and a manager at Johann Friedrich Blumenbach Institute for Zoology and Anthropology, where he also leads a lab at the Department of Cellular Neurobiology. His lab has several major interests. One of them is the neurobasis of insects' behavior, with focus on sex-specific mating and aggressive behaviors of acoustically communicating grasshoppers and fruit flies. In addition, Professor Heinrich is curious about the neuroprotective function of the molecule erythropoietin and its role in neurodegeneration. Another intriguing aspect of Professor Heinrich's research is the process called apoptosis, which is a form of programmed cell death that occurs in multicellular organisms. Before we begin, I'd like to mention that this podcast is separate from my research position at the University Medical Center in Göttingen. This podcast is supported by the International Max Planck Research School for Neurosciences in Göttingen, as well as by the Cluster of Excellence Multiscale Bioimaging in Göttingen. And now, please enjoy the talk with Professor Ralf Heinrich. Hello, Professor Heinrich. Welcome to our podcast.
Yeah, thanks for the invitation.
I'm glad that you accepted our invitation. Usually we start with a short introduction of our guest and how did you ended up in science. If you can please explain and tell us your story about this.
Mm-hmm. Yeah, so I was born in Bremen and then I grew up in a very small town near the Dutch border in the north of Germany, and end of school time I really didn't know what to do as a professional career later, and since I had a very enthusiastic biology teacher in the last years of school who brought in lots of experiments and all these things, I thought that's the thing that I liked most in school. So I started to study biology, and I did this in Marburg. Also focused then a little bit already on neurobiology, then came to Göttingen for a PhD, where I mainly worked with neuropharmacological methods in the brain of insects to study the selection of appropriate behaviors and things like this. After the PhD, I went to Harvard Medical School on a Humboldt stipend and spent 2 years as a postdoc there, working on aggressive behavior of lobsters and crustaceans in general. And then I came back and received one of the, by that time, newly invented junior professorships, which I maybe for the first time gave young investigators full responsibility right away in terms of teaching and research and examination and stuff like this. After that, I had a lot of short contracts until finally in the end I received a permanent position here. First as an assistant professor in a certain department, and since a few years I'm more, it's called the manager of the institute here that kind of does some administrative support for the director, basically.
Mm-hmm, mm-hmm. And is there any particular reason besides the job offer that you got to come to Göttingen? Or this was just, at that time there was this offer there and I was already there, so I decided to come back?
Yeah, I think that was already a reason, because during the PhD studies I really liked Göttingen as the place. I kind of remained in contact with my doctor father who supervised my PhD, and therefore this connection was always established, and it was kind of, a little bit also planned right from the beginning to maybe come back to Göttingen if there is a possibility.
Mm-hmm. So you already had this experience, nice experience here, and decided, okay, let's go back there. I was happy there. and it turned out to be a great choice, I guess. You're
Yes,
still here.
in the end. No, I really like it, I have to say.
So you mentioned you was in Harvard Medical School, and last, I don't know, 3-4 guests, all of them, they went to the US. And I'm starting to get curious. Is there a reason to people to go exactly to US and then come here and become leaders, or wasn't it popular, for example, to go in other country in Europe or China, for example? I don't know. And this is the first question. The second question is maybe again to make a comparison. We ask also others about the research there that you did at that time and then the one that has been done here. What are the differences, similarities maybe?
Yeah. So we went to Boston to Harvard Medical School in 1997 till 99. So this is almost 25 years ago. And by that time, I think it was the typical thing that you went for the United States for a postdoc and then come back to Germany or even stay there and take a position there. I would think that today it's not so important to go to the US, but I think it's still a good thing to do a postdoc period abroad in a foreign country, working on a different subject, learning different methods, and trying to get research done and good results also in a different environment. But as I said, I think it's not so important anymore to go to the US. You can choose any other country.
Mm-hmm. It's important to be outside of Germany.
Yeah, I think just to show that you can work and be successful and creative in a different environment, basically. Mm-hmm. Concerning the comparison, again, it's 25 years ago. I experienced the daily work of research very similar as to the situation here. So I had the fortunate situation that I had a PI there who was kind of a team player, not above the group, but within the group. So we were doing research all the time together, and he was interested in experimental results and research results as much as in personal relations, so that everybody felt fine and things like this. And I really enjoyed this atmosphere there, which was very creative and motivating to contribute whatever you can give to this group. Mm-hmm. What was the case at that time, that teaching was really completely different. So I also taught a neurobiology course at Harvard University, which was quite interesting because we had these tutorials and very small group intensive supervision of the students throughout the lecture series and all these things. And we didn't have this at all in Germany by that time. Now you have this, for example, in the master programs here, but still at the university with the bachelor programs with 100 or 120 students starting each year, we still cannot do this in this intensity. But this was quite an interesting experience, and I really thought that it's a helpful thing to educate the students.
Mm-hmm. Indeed, it is very helpful. And for the people who don't know you that much, you are part of our master graduate program, International Max Planck School for Neurosciences, and you're heavily involved there in the program committee, in the teaching also. And I enjoyed your course, by the way, quite a lot. It was quite fun. And I had the feeling that you also enjoy a lot being around students. I wanted to ask you, what is it for you to teach? How does it make you feel?
So for me, teaching is really at least half, if not more, of the job that I have to do here. I mean, and teaching includes giving lectures and courses and all these things, and I really enjoy to make up certain topics and adjust them to different audiences. So, for example, I participated in the Children's University 3 times so far, but it also is in giving lectures to students on different levels, and also I do my research usually with the students. So I really enjoy to be part of the team and try to motivate them to contribute, and we discuss mine and their failures and successes in a most informal way and stuff like this. So I really like to interact with the students, and that's basically it. And also concerning your program, this IMPRS program, I also like to participate in your PhD retreats and things like this. And even if I'm a professor, I get it. It's always a bidirectional way of teaching. I mean, participating in these retreats and lectures and seminars and stuff like this also gives me a lot of information and backfeed and things like this. And I also have to say that, for example, with my last 2 PhD students, I'm sure that I learned more from them and with them during this period than they learned from me during this time. So it's really, a lot of fun and rewarding and motivation that I get from these interactions with the students.
I think also the interaction with the students and this, how do you call it, this thing that you, basically there's no distance between you and your student. You're like more like co-workers in the lab than you're his or her boss. And this also contributes a lot to the learning process, and people feel more relaxed and they are more open to share with you some difficulties so that you can help them. And honestly, I wish that more PIs are like you and go and behave that way with their students, so that also they're showing them a nice example. And when they at some point eventually become PIs, they will also perhaps treat their students that way. So, which is a win-win situation to everybody.
But probably it's easier for me because I usually run a very small group, and I'm usually doing research with bachelor students and master students. Typically, I only have one PhD position filled, so it's not a huge lab that I have to organize and get running with different projects in parallel and things like this. So I really have the time to sit with the students, do experiments together with them, and maybe if you are more advanced and on a high level involved in university or MPI structures, you don't have the time to do all this.
Yeah, that is correct. This is a fair point. Indeed, people are extremely overwhelmed with tasks, and they're overcommitted to different activities. However, it would be cool if they, I'm not judging anybody, right? They are doing an amazing job, people here that I know at least, and also others. But maybe if you can assess yourself and your workload, and then say, okay, this time I cannot supervise 5 PhD students, I have the time only for 1. This is completely fine. I am not aware of, I don't know, maybe they do that, I don't know, OPS, but teaching is definitely crucial, not only to your students, but also not only to the students in the lab, but also to your audience. When people at the university have some lectures, etc., this is also very important. I'm changing slightly the topic, but you are one of the main local organizers of one of the, maybe the biggest neuroscience
in Germany.
In Germany, the biggest conference for neurosciences in Germany, which is organized by the German Neuroscience Society. And I don't know for how many years have you participated here, but you are always there and work on the background. And I would like to ask you to share with us what is this giving you to organize such a big event and be part of it for many years?
Okay, so this meeting is quite a traditional one by now. I think it has happened almost 40 times so far. And actually, when I came here to Göttingen for the PhD, the group that I joined of Professor Elsner, he was the local organizer of this. So I really have memories of the whole group being involved in printing posters, setting up the poster walls and all these things. And by that time, there was no digital stuff. We really did it all by hand, by ourselves. So by that time, it was really a big issue, and we had to invest a lot of time for this. Nowadays, the Neurowissenschaftliche Gesellschaft or Neuroscientific whatever society, they do most of the registration. There is a panel that sets together the scientific program and things like this. And then I'm more the local coordinator of the on-site events that have to be done. And then we receive a lot of help from the different institutions here in Göttingen, from MPEs, from the DPZ, from the ENI and the medical faculty. So there are helpers that are also experienced by now because they did it several times. We have all the materials basically at hand, and also the procedures of setting up things and stuff like this are quite established meanwhile. So I would say my task is to coordinate these things in the 2 weeks before until the week after this meeting, and all in all, I would guess that it costs me maybe 3 weeks of full-time work to get this task basically done. But as I said, without this work that the Neurowissenschaftliche Gesellschaft is doing there, we wouldn't be able to do and handle such a big conference by our own anymore.
It was a great experience. I attended this year actually in March, and I don't know how many people, nearly 2,000 I guess.
Yeah, between 1,500 to 2,000 in the strongest years. And this meeting is still the one that kind of supports the young students to come to Göttingen. They have low fees basically for the young students, and for many neurobiology students, that's actually the first meeting where they start to present a poster and talk to the big guys in the fields and things like this. And also, since this meeting has been in place in Göttingen for such a long time, there are basically boundaries to all these different groups, so everybody knows somebody here, and people meet again, and they sleep at different colleagues and friends from former times and stuff like this. So it's always also a reunion of old friendships and colleagues and labs basically.
It's fun. I also was able to present something, my first talk in such a big event, and I remember I was pretty nervous, but at the end it was fine. I mean, it's a good practice to have this such events. Do you know why did they choose Göttingen for the place for the conference?
I think it started at the Fassberg Max Planck Institute. And it started as a very, very small meeting, which was not openly announced, just a gathering of some colleagues from some other cities or even countries, I don't know by that time. And then they decided, okay, we should do this more often and on a regular basis. And then it was moved down from the Max Planck campus to the university campus and grew over the years, basically.
Again, it started in a small circle. Very interesting, very interesting. And now it's like, I guess, 20 plus years it exists, the society.
Almost 40, I think.
Almost.
Yeah. I think the next one is the 40th, if I remember correctly.
40th. Oh, wow. Then it's longer than I thought. I was confused with some other event. This was the Lange Nacht der Wissenschaft, I think, the long night of sciences, where people open up their labs for general public and And everybody is able to visit the lab and ask questions about research, which is also a great
this is
initiative.
a great thing, to get some insight behind the scenes and participate in activities and stuff like this.
Exactly. Okay, so we know a little bit more about you and your motivation to do science and the teaching. And now I would like to go into the direction of your research and ask you, what is your research topic?
So I have shifted my research topic actually several times during my career, and the actual one is basically the evolution and function of cytokine signaling. So cytokines, to explain this, are small molecules that regulate processes like immunity and inflammation and the development and specialization of any cell types, basically. And we study a certain type of receptor, which is called the CRLF3 receptor. And this, we believe, is the grandfather, basically, of all these cytokine receptors that we have now, for example, in the humans also. So.
And you use insects for these purposes? As long as I remember, grasshoppers and fruit flies also?
No,
not anymore. Okay,
half of them. We are using insects. As I said, this is one of the evolutionary oldest molecules, basically. So we know that it appeared with the appearance in evolution of the first typical nervous systems.
When would it be?
I don't know. I don't know. But even the polyps and the cnidarians, as we call them, they already have this receptor, and it has been stable, and the same gene basically and molecule exists throughout the entire animal kingdom, including worms and crustaceans and insects and all the vertebrates until the human itself.
Cnidarians, these are the jellyfishes, right?
Yes.
Yeah, exactly. They are very interesting, and they have no sensory system, but some kind of nerves there. It's very interesting.
I mean, they have a nervous system already. They have typical neurons that rely on the same functional molecules as our neurons in the brain. So since the neurons evolved, so were established very early in these ancient creatures. They were already in place in the last common ancestors, for example, of us, the mammals, the vertebrates, and the insects, for example. And therefore, all the functions that were already established in these the common ancestor creatures, they are basically the same or very similar between, for example, us and an insect.
This is maybe an answer about the next question that I wanted to ask you, but maybe you can elaborate. I wanted just to talk about how this primitive, on first place, at glance, so to say, creatures like insects, fruit flies, or grasshoppers can give you insight about complex processes such as neuroprocessing and connections in the brain. And why do we study these insects, for example?
Yeah. Indeed, that was the beginning of the answer already given. Since we have this common history throughout evolution, at some points this is estimated 650 to 900 million years ago. There was one creature that was the common ancestor of all the insects and also the vertebrates, including mammals and humans. So everything that was already established in this creature is basically the same in all these different animals that evolved from this ancient creature, basically. And it's already known, or it's known that this basal creature that we call the Urbilaterian had already a nervous system. It had a complex nervous system with different regions dedicated to particular functions. It had sensory systems and sensory organs that picked up information from the environment and which was then processed in the central nervous system and things like this. So it had neurons that had the full repertoire of functional molecules that make up the characteristics of a neuron. And since this common ancestor already had all this, we can basically study mechanisms related to these functions in an insect as an equivalent also to a brain cell from our brain.
Mm-hmm, mm-hmm. So basically the similarities very conserved, highly conserved, and you said you can use
so
this.
That's the principle in evolution. Mm-hmm. Things that work well will be kept and a little bit elaborated on, but as long as they are useful and necessary, they will be taken to the next level in evolution, if we want to say so.
Mm-hmm. Could you maybe compare the brain of a vertebrate, let's say human or a mouse, and a brain or a nervous system of a grasshopper, for example?
Yeah. I mean, the first difference, or the most striking difference, is of course the number of neurons included in a vertebrate or mammalian brain compared to an insect brain, which is, I don't know, an insect brain has 10,000 to 50,000 neurons, for example, depending on the species, and we have billions of neurons in our brains. But the principles of information processes by the neurons, the ability to transmit information from cell to cell, and also the ability to perform complex behaviors or functions with this, like learning, adapting your behavior to certain situations, forming long-term memories, and all these things, is based on the same molecular processes in all these creatures, basically. Mm-hmm. And therefore we can use or study these basic functions in an insect cell as well, as an equivalent also to mammalian brain cells, for example.
Mm-hmm, mm-hmm. You mentioned neurons. Do they also have glia cells, these supporting cells in the brain?
Yeah. So these invertebrates also have glia cells, but they are not directly characterized in these three large groups that we have in the mammalian or vertebrate brains. Which fulfill certain functions, but you find equivalent functions for these vertebrate-type glial cells also in the invertebrates. So we have a blood-brain barrier that separates the central nervous system from the circulation and the rest of the body. Mm-hmm. We have insulating glia. We have glia that supports the neurons with energy and metabolites and stuff like this. So glia is already there, but it's a little bit different from the types that you can assign them to than the vertebrate glia.
Mm-hmm, mm-hmm. And what kind of methods do you use usually, or maybe throughout your career? You mentioned you switched topics.
Yeah.
You can mention some of the methods and tools that are suitable for insects, neurobiology, for example.
Yeah. So for the actual studies, we usually take brain cells into culture. So we basically dissect out brains of locusts, for example. We dissociate the cells, so we rip them out of the brain tissue, and then we attach them to glass slides, basically, and put them into a sterile culture medium and let them grow for a while. Mm-hmm. And then we basically test whether cells can survive certain challenges better or the same or worse if we treat the cells with certain drugs, for example, or certain molecules. Mm-hmm. So what we typically do, we set these cultures under oxygen shortage, what we call hypoxia, for a certain period, and then we compare the survival these hypoxia treated to untreated cells and hypoxia treated cells in the presence of a potential neuroprotective substance or a toxin or things like this. And by this we kind of can find out whether certain substances help the cells to survive these harsh conditions, the lack of oxygen.
And for example, when you find something in these cell cultures, do you then go back to the living animal and try to apply some drug or some stimulation, inhibition in the living animal?
Sometimes, yeah. But that depends on whether we can find a readout for this. So, for example, if you want to stimulate adult neurogenesis, you can mark these newborn cells also in an intact brain and later label them and count them and see whether a certain drug has an impact on the proliferation, the generation of new neurons, for example. Or you can use sometimes a behavior as a readout. So, for example, in one of our first experiments where we found these protective functions of erythropoietin in insects, we were able to just crush the nerve of one of the ears of a grasshopper and then use an acoustic orientation behavior to monitor whether these neurons regenerated and re-established the hearing ability, basically. So and this is also a advantage of invertebrate or insects as experimental animals, because they tolerate this being captured, being immobilized much more than mammalian species, and they still perform their somewhat natural behaviors and reactions under these experimental situations.
And also, for people who are not doing science in Germany, as long as I am informed, insects are not seen as animals. And therefore, you can perform genetic experiments and many different sorts of experiments without needing these permissions, these heavy administrative burdens. For example, we work with rats in our lab, and we do the same. We also dissociate the brain. We grow cultures, but if we want to then see how this process that we found out in the cell influences the living animal, then we need another permission, and this is another half a year to year, and it's very, very hard to do this kind of research.
Yeah, in this respect, it's very easy and comfortable to work with insects because, as you said, we don't need specific permission for each experiment that we are planning and doing. We can order these locusts, for example, because they are reared as food for reptiles and all these things. So there are commercial breeders that send us these experimental animals by mail each week. So that is not a problem. And there is not this ethical problems because I wouldn't say that insects are not treated as living beings or something like this. They also react to being grabbed, being taken, and stuff like this. So even if you work with insects, you should think about how long they have to be alive and whether you can sacrifice them in a way that they don't feel this panic-like reactions that every being basically experiences when it's in danger, basically.
Of course, once the creature will experience some stress, for example, which is very bad for it, and also this might influence your experimental outcome. So nobody wants to stress out animals and do harm to the animals. And we apply to the 3 R's and reduce, replace, and the third one I always forget. Reduce, replace. What was the third one? Refine. Yes, refine. Oh my God. But so indeed in these animals it's a little bit easier, and also you can even see some of the nerves, right? With the bare eye. I mean, I don't know in locusts, but although You can see them. Yeah. They are accessible, and it's nice to do electrophysiology, for example.
Yes. I mean, invertebrates sometimes have extremely large diameter neurons that you can record from, especially for learning electrophysiology. It's very rewarding to record from a leech neuron or so, because you can hold this connection to the neuron with your sharp needle for long times and things like this. But if you want to get information about the information processing in an insect neuron, it's hard, because you have to go into the dendrites or the axons. Since the neuron is a little bit differently structured as the vertebrate brain neurons, if you just poke your recording needle into the cell body of the cells, you may not see any activity, because it's not on the pathway that the information is basically relayed from cell to cell.
That is interesting. So there is some different organization in the structure, and also do they have the same neurotransmitters as in humans, for example, or vertebrates?
Yeah. So most of the major neurotransmitters are the same in the insects. We of course have a lot more like neuropeptides, which consist of series of amino acids and things like this that are very species specific. That you might not find in the insects, but in general, all the major transmitters and also the receptors that react to these transmitters are also established in the insects. So all this obviously also was established already in the Urbilaterien, which was the common ancestor of us with the insects.
It's just fascinating that it kept going for so many millions of years. Yeah. And we're talking about millions.
Hundreds of millions.
Hundreds of millions, exactly. Wow. And it's still the same, so meaning that this is indeed the most efficient way, for whatever reason, to sense and transform information, and it's just incredible. You dropped the term erythropoietin.
Yes.
And that is a ligand for these receptors that we're using, the cytokine receptors. Yeah. Could you please elaborate, how is this molecule, erythropoietin, important in the brain? As long as I know, it's produced by the kidneys and liver, and it stimulates red cell production. But what is the role in the brain of this molecule?
So the name erythropoietin is coined because the stimulation of red blood cell production was the first function described for this molecule. And as you correctly mentioned, it's typically released by the kidney in situations when there is a lack of oxygen in the tissue, and the more erythropoietin is built, then more red blood cells will be produced, and these red blood cells carry the oxygen in the organism, and therefore we have a better oxygen distribution. And the erythropoietin in these blood production system basically prevents the degeneration, the cell death of the developing red blood cells. So developing red blood cells without the EPO signal will undergo self-destruction, which we call apoptosis. It's a regulated program that kills cells, and therefore only if there is an erythropoietin available and activates the receptors, these cells will be mature cells that can really transport the oxygen in the organism later. And basically later people have found that erythropoietin is also produced and released in other tissues, including the nervous system. And the stimuli that lead to the production and release of erythropoietin in the nervous system are very similar. So there's also hypoxia, there is toxins, inflammation, things like this, mechanical injury, but it's also high neuronal activity. So also in terms good activity related to social enrichments and all these things. So whenever there is local high activity in the brain. It seems like erythropoietin is produced and acts on neighboring cells that express a receptor for erythropoietin. And the typical erythropoietin receptor that we have on our red blood cell, that's a molecule which is very characteristic, and it's also present in the nervous system. But we as humans and other mammals also produce also a variant of the erythropoietin molecule, which cannot activate this receptor, this specific receptor, but can still mediate protective effects on nerve cells. So in order to achieve this, there must be another receptor to distinguish between these two functions. And this was basically the idea why we started with the work on insects, because insects don't produce erythropoietin. Only with the evolution of the vertebrates, all these signaling substances evolved. So insects don't have this, but still the human erythropoietin can save the insect neurons by activating the same pathways that are activated, for example, in our brains.
This is, I don't know, it's surprising. It's kind of inverse evolution. I don't know. How did you found this receptor eventually?
Yes, and actually you mentioned Drosophila.
The fruit fly?
Yeah, and Drosophila helped us by finding this receptor because it doesn't have this receptor. Yeah, so when we started our experiment with the locusts, we saw this effect. The neurons were protected with erythropoietin, and then we went into Drosophila because Drosophila is the best researched insect. You have the full the gene sequenced, and you have all these sophisticated methods to manipulate this Drosophila. So we thought, okay, let's establish this neuroprotective functions in Drosophila, and then we work with that animal. And we burned a few bachelor and master theses on on this project, but there was no effect on Drosophila. And then we found a publication where genes were compared between different animals, just in general, how similar are the genetic repertoires of certain animals. And there they mentioned a CRLF3 receptor, which we didn't know before, which was lost in Drosophila, but was still present in more typical, more basal insects like locusts and locusts and crickets and these type of animals. And then we thought, okay, that this receptor is kind of similar from its structure to the erythropoietin receptor of the vertebrates and to thrombopoietin receptor and all these blood regulating production or blood production regulating cytokines. Let's give it a chance and try to see whether this is probably the receptor in the insect. And indeed it was. We were able to show this. But the interesting thing now is we don't know the natural ligand in the insect. So we know the receptor is there. We can activate it with human EPO, erythropoietin. We can activate it with this splice variant that does not activate the specific receptor in humans and vertebrates. We can activate it with thrombopoietin, which is a different cytokine involved in platelet production, the blood cells, and but we don't know yet the natural activator of this CRLF3 receptor in the insects. And since the signal binding properties of these receptors have been very similar so far between the insects and all the mammalian cells, including human cells that we tested, we would expect that this unknown natural transmitter in the insects could also activate the human receptor, which would be an interesting substance to test in terms of preventing neuronal degenerative cell death and things like this.
That's indeed a huge potential for this purposes. I mean, it's very interesting when you say it. Like, in order to prepare for this podcast, I read a little bit also about your research, but when you explain it like this, it makes a lot of sense, and it's very interesting to understand now, okay, why in first place do they have this, and as you said, what is the ligand and how it will eventually influence vertebrate cells. I just want to clarify here one term. You mentioned splicing. This is basically when the cell reads a gene, this gene it's being processed, and because of this processing, for example, you can have two different proteins that are very similar but have slightly different function, and from the same gene, for example. And this is more or less the splicing. Of course, it's way more complicated, but for people who want to read up, they can go and do that. When you say neuroprotection, what do you mean by that? By this neuroprotection of the cell, what is meant by this?
Okay, so when typically if there are normal conditions, let's say like this in the brain and in tissues, the cells survive, they maintain their functions, and they are happy in a way, and yeah, as I said, but then conditions might change. The cells might be challenged by, I don't know, infections or damage or things like this. And under these conditions, sometimes these cells decide to activate their self-destruction program, to eliminate themselves from the tissue in order not to harm the neighboring cells. And in this case, for example, neuroprotective substances can basically inhibit this self-destruction program and let the cells survive even under this non-optimal, suboptimal conditions, basically. And this is how many of these neuroprotectant substances act, that they kind of block this self-murdering apoptosis mechanism in the cells.
Why did in first place apoptosis occur? Do you know that?
I don't know. I mean, it's a process that happens all the time, and it makes sense in a way that kind of compromised cells or infected cells or cells that are produced in too many numbers are eliminated, and in contrast to just degeneration of the cells, where the cells kind of explode and release all their ingredients into the surrounding tissue, which harms the neighboring cells. This apoptosis program is a way where the cell basically silently dies without affecting any of the neighboring cells. And I think this is especially important if cells are infected. So to kind of keep this infection into the cells and not let it spread to other cells. That's basically the thing. And we have certain tissues, for example, all our gut systems, our olfactory systems, where we have a constant. The skin, of course, where we have a constant replacement of old cells which die by apoptosis against new cells that are generated from certain stem cell regions that replace these old cells all the time.
So it's a cell death, but it's something that is good in the sense of the organism. It keeps it going.
And it's a very regulated process. It has some genes that are necessary for these activating factors, and for the series of events are specifically activated under certain circumstances to eliminate a particular cell.
In your research, as long as I remember, you also dealt with this topic, the apoptosis, and there is something about acetylcholine esterase, right? Which is one enzyme that degrades acetylcholine, one of the neurotransmitters in the brain. And what was the influence of this molecule on apoptosis?
Okay,
the relation?
So it has been found in many mammalian cells, and we also found this in insect cells, for example, that these conditions that drive the cells into apoptosis, into the self-destruction program, they also lead to an increase in the presence of this enzyme acetylcholine esterase. And if you just block the function of acetylcholine esterase, cells will survive also harmful conditions. So we did this with this experiment that I described already. We put cultured brain cells into a hypoxic chamber. And if we do this in the presence of a blocker, an inhibitor of this acetylcholine esterase function, then the cells survive this lack of oxygen. So this acetylcholine esterase seems to be a important functional step in this self-destruction program in the apoptotic program.
But just to interrupt you here, could it be also acetylcholine? Does it play a role? Because when you don't have the esterase, you have more of this, let's say, bound or released. Is there any role in this? Acetylcholine, the neurotransmitter?
That's not really clear. I mean, there are different ways how acetylcholine esterase may contribute to the cell death program, basically. So it has been found in certain cells that it is participating in a huge complex that activates the enzymes that execute the final steps of cell killing. Then it has also been found that it, under this deleterious conditions, invades the nucleus and acts as an enzyme that destructs our DNA, which contains the genes. And it also, this indirect way that you mentioned, that there is less or more acetylcholine if the esterase is less, and vice versa, can also happen because it has also been shown that activation of the receptors for acetylcholine may contribute to the well-being of the cells, basically.
So it's a complex story. It's not one way or the other.
Yes.
As usual in biology.
Yeah.
So the esterase. Okay, interesting. Why? I don't know. Why is not the right question in biology, I think.
but there are lots of questions connected to this. I mean, as you said, acetylcholine esterase is typically found in the synaptic gap, or the gap between two neurons that communicate with each other, and it's produced as a protein which is exported out of the cell. It has all the signals that it gets basically transported to the cell surface and released in the environment. And it's totally unclear why acetylcholine esterase appears in the cytosol to perform these other functions that I described in apoptosis and other cellular functions. So there are many basic things which are just mysterious still and have not been really studied.
And also something interesting, there is this acetylcholine. There are two different type of receptors, and acetylcholine acts differently on these receptors. For example, we have it in the heart, and we have it in the neuromuscular junction, right? And I don't know. The insects also have muscle, right? We studied muscles in insect, and I remember this.
And what was the transmitter?
This was it, GABA?
Also.
GABA and acetylcholine.
And glutamate.
And glutamate.
Not acetylcholine.
Not acetylcholine, exactly. See, I forgot some details. And he was one of my examiners during my exam, the oral examinations. I don't know if you remember, but it was quite fun. Yeah, so we covered some of the topics, and now there is another one that you are interested in, at least according to the information on the webpage, and it's about the social behavior of insects in terms of aggressive behavior, sexual behavior, and auditory information processing. First question would be, how do insects hear, actually?
They have, depending on the species, quite different mechanisms to do this. So we have mentioned Drosophila, the fruit fly. They basically hear with their antennae. So antenna is moved by the pressure differences, and then this is picked up by sensory cells, these movements, and they get excited and send this information to the brain. Other insects have their ears in the legs, in the butt, the abdomen, or at the wing bases and things like this. So depending on the species, it's quite different mechanisms and sensory organs that insects can use.
But is it mostly mechanical transduction or something else?
Yeah, it's of course mechanical transduction, where there are typical sensory cells that are associated with something that is vibrated or moved by the sound waves, basically, and this is picked up as a mechanical signal, and then changed into electrical signals and sent into the nervous system.
And what exactly is your focus of research in this regard, in the behavior of insects, for example?
So actually this, we are almost not doing this anymore because the focus was completely shifted to this erythropoietin and CLF3 project. But yeah, I mean the insect or invertebrate behavior is also kind of a way of regulating the progress of evolution and regulating which animal is the preferred reproduction partner and generates offspring. And so all these behaviors, like the acoustic behavior, which is kind of used by insects to find mating partners, sometimes to assess the quality of a mating partner and made the selection with which one I will mate and generate offspring. And it's also the aggression behavior is used to kind of establish who is the most dominant animal, who usually then gets the right to reproduce with a conspecific. And then the interesting thing in these competition within species is that although they could harm each other and kill each other, the behaviors are sometimes or very often ritualized. So taking the lobster or taking, for example, poisonous snakes and things like this. I mean, these snakes could bite each other and kill each other, and if they bite at the same time, they will both die. And in nature, if you are injured in such a fight, it's basically you're sentenced to death because you get infections and all these things. So you have to avoid to be injured, but still you have this competition where you want to establish who is the strongest, the best, and gets the preferred reproductive ability or no ability alone is something like this, and therefore they have ritualized their behaviors, that they don't use these deadly weapons to kill the other, and they kind of go in a series of increasing intensity of aggressive behavior from just shoveling, testing the strength of the other, and then it's kind of a wrestling, and then only if none of the opponent retracts on this low level where you can't be injured, fights escalate to intensities where there is the potential to be harmed and compromised. And even if you are the winner, if you take away some injuries, you might die later. So nothing is won in this. And how this is regulated and how this is, since the animals usually don't learn this, so it's somehow imprinted in their nervous system, in the development, whatever. How this is regulated is an interesting thing.
So basically, you can observe this behavior of the animal, and is it possible to backtrack? Okay, now we have this behavior, these neurons were activated, this was released, and then draw conclusions and test hypotheses if certain set of neurons is active during certain behavior, and vice versa. Can you manipulate, for example, these neuronal pathways to make the animals exhibit these behaviors?
Yeah. So there are certain transmitter or hormone systems which are throughout the animal kingdom connected to this agonistic or aggressive behaviors. So serotonin is involved, and octopamine or norepinephrine, which is the equivalent in the vertebrates, and even during fights where animals already have decided to retract from the fight because they gave up basically and they want to avoid to be injured, if you boost the system of them, for example, inject serotonin or so, they immediately resume fighting. So it's all you can really pinpoint activity and aggressive motivation and all the complex behaviors related to this to certain neurons and their activity and the release of the signals that they generate.
So it's interesting in terms of free will. I don't know to what extent animals and insects have this will, but it's quite interesting that one neurotransmitter triggers you immediately and puts you in completely different mode, and you start
but
behaving.
I think it's not different in us. I mean, if you also think of depression and anxiety disorders and all these things, I mean, boosting a little bit, very generally, the serotonin system changes this situation completely.
Serotonin, dopamine is very important in this kind of behavior and reward. And yes, but still, we still think that we have free will. I mean, this is a very philosophical question. I mean, biology is complex and fascinating. So we covered quite a lot of scientific subjects and topics which were very, very nice. And now I tend to go to the not so nice part of the conversation and talking about problems in our field. And you have been already for many years in this scientific community, and you know very well that there are plenty of problems that we need to solve and maybe somehow make the scientific machine works better. And in the previous episodes, we talked about the fact that many times people from one lab struggle to reproduce experiments that have been done in another lab, for example, in different location in the world. Also, there are highly skilled people like postdocs, PhDs who are trained, and then instead of staying as a postdoc in some lab, they decide to leave academia, go to the industry, which is totally valid and sometimes even encouraging. But in this way, the scientific community, so to say, lacks this kind of skilled, trained people. And this is seen as a postdoc crisis, for example. And there are many, many others. For example, the fact that, as you very good know, people who do research, they need to teach, they need to do administrative tasks, they're involved in many committees and etc. And their time is basically, I don't know, how do they do it, especially directors of institutes such as German Primate Center or Max Planck's. And in addition to that, we have the publishing industry that it takes a lot of time to accept a paper, and the reviewers very often they don't give valuable feedback, but they just destroy some work. And basically you've been working on a project for 2-3 years, you submit your paper and you wait for another year to be published, and at the end someone says that just doesn't like it and it's not accepted. And this will also maybe hinder you in getting a funding for your future research. And all of the things are connected. And of course, it's not that bad as I describe it. I mean, these are problems, but still the system is functioning. However, we can do better. And if you pick up some of these topics, maybe others, and give us your opinion on that, and maybe some advice to us and to all young people who listen, what can we do and what can be done in order to improve this system?
Mm-hmm. Maybe starting with this publishing issue. I mean, it's clear that whenever people are involved in deciding about things, it's not objective. Mm-hmm. And if you send in a paper to a journal, it's a matter of luck whether you receive an editor who generally likes the story and is interested in this topic. So then once you pass this bodyguard at the entrance, mm-hmm. you're already halfway through. And the same with the reviewers. I mean, if they really like the topic in general, they look at it in a different way than other topics that don't really bother them, and then there are bad reviews. I agree sometimes, but my experience is that even if you receive critical reviews and you are a little bit angry about this in the beginning, to do the extra work and do it again and read it again and rewrite things and do some additional experiments even, or stuff like this, in the end it makes a better paper. And
just to interrupt you, yes, that is true. This is the idea, to make the paper better. But in many cases there are this just personal opinion. I don't like it, and that's it. And So I don't know. Maybe a way to get out of this is to make also the reviews available. Mm-hmm. So for example, with these frontiers journals, they at least disclose the name names of the reviewers once the paper is published. And I think this is something that kind of forces a reviewer to put a little more effort in his or her review, and also be able to defend why this judgment has been given. So I think that's one thing that I like about these new developments. But you're right. I mean, if there is a reviewer, especially in a high-ranked journal, or even if you apply for research money at certain foundation, if there's only one of 3 reviewers who is not overwhelmingly convinced of this, you're out basically. And this one person can be wrong with the same probability than all the others. So in a way, it's of course not fair. Yeah, and it's in principle anonymous. But everybody knows the work of the others, and people tend to cite their work in their paper, so it's very easy to find out who has written this. And it's also sometimes conflict of interest, that these guys, no, I will not let them pass. I don't know.
Yeah,
it's a hard. I think eLife they have this that eLife, was it that reviews are also uploaded? I'm not sure. I might be mistaken. Yeah. So they
maybe
say
that's why it takes so long to publish.
It's a huge file. Yeah. So this was one of the points.
Yeah. Another point you mentioned that researchers don't follow an academia career, but leave for industry and other jobs. Why not? I mean, we are at the universities. We always focus on educating researchers, but if in the end 5% really end up in their future profession as a researchers, I think that's a quite good estimation. So probably in our education we should shift the contents and the topics a little bit more to applicable things and skills that one can also use in other professions. If you want to go for an academic career, I think the difference between academia and industry is the predictability of your future. If you go into an industry company, you get your task. If you do your job reasonably well, and then you can keep your job or you get promoted. Once you have gathered some experience on your job, you become increasingly rapidly very valuable, and you can switch between companies, which seems to be quite easy at the moment, and do this game until you are in a position where you feel satisfied and you like it and you stay there. So if you do your job well, you know that it goes forward and you can make a living out of this. In academia, that's different. So the first steps are still predictable. You study, you do a PhD. Getting a postdoc is also still quite easy. But then there is this next decisive step where you either get a young researcher group somewhere, or you join a department and become an assistant researcher or something like this, do a habilitation, and then apply for professors. And here that's the problem. It decides very, very late, at least in Germany, whether you can make it to a permanent position or not. And even if all the steps before were successful, you publish regularly, you do good teaching, you get third-party money and all these things. If you're not at the right place at the right time, having the right people making the decision, you will not get a permanent position. And this is the risk. So in my view, it's a smart decision to leave if you want to have a predictable future career. And if you decide for academia, you should be aware that it can end in a dead end, and you have to, after a while, switch completely to completely different things. And you should only do this if you really enjoy this research and the way of working on such topics. And if it doesn't lead to a permanent position, you should be able to look back and say, I had 10 really great years and I'm not regretting anything, and now I start something new, and don't see this as a failure or a waste of time of my life or things like this.
Interesting. So if you're not passionate about, leave earlier, and then you will be happier than struggling. And as long as I know, it's even harder when you're in the system for 5-10 years to go to the industry. People in the industry tend to welcome PhDs that immediately after their graduation go to the industry, so that they can gain experience in a different environment. And
I'm also a good example for this. I mean, so both in a positive and a negative way. First of all, when we were in the States, my wife, she studied human biology, and while we were in Boston, she was able to enter an institute and get into clinical studies a little bit. And this gave her the opportunity, when we came back, to immediately finding a job, a real job, a secure job, leaving me the space to do this insecure path and follow a career in academia. But if you completely rely on the money earned by this unsecure and unpredictable future, basically, it's hard.
It's tough, yeah. So scientific families are
Yeah,
not
yeah.
encouraged. At least one should be out.
Yeah, one of them should have a real job and the other one can play.
Yeah, exactly. It's exactly the playground with a lot of money and people involved. Yeah. So this would be the second one, the security and postdoc living. Is there anything else that you would like to share in this regard with the problems?
I don't know. You also mentioned work-life balance or something.
We can talk about this. Yeah.
Yeah. I mean, I think it can be rewarding to really work hard and long hours for a while, especially if you work in a team, and that's really great to get things done. But you cannot do this for years. I mean, you have to find your own pace. You have to have hobbies. You have to have social life, and you have to take your time to keep these things. Otherwise, on the long run, you will lose motivation for research, and even worse, you get sick or get other problems out of this. So, but what is too much, and what you can still stand without losing fun and motivation, that's very different for individual persons.
Yes, indeed, there is something personal, and everybody is different threshold of stress, of working load that they can handle, workload. It's definitely. And there are some people who actually seek this, but there is this thing that at some point, as you mentioned, you cannot do this forever. Yeah. You are burnt out. And now people start recognizing this condition when you are overworked and you need a break somehow. And then when you have the break, you're again more productive and you can do things better and faster.
And then, of course, it helps if you really do things in your profession that you like, then they don't feel like, I don't know, work and things like this.
Exactly. In my case, so far, I don't feel like I'm working.
Yeah.
Although I think I do fairly a lot, but I'm thinking about the future now, right? And when I start thinking about all of this, I don't know, broken system to some extent, do I want to do that really, or should I switch some? We'll see. This is something personal, and we will decide in 2 years from now, at least I and some people behind the cameras even sooner.
Yeah, but it all depends also on the other conditions of your life, whom you met, whom you meet, where you go, and things like this.
Family and etc. Yeah. Maybe now is a good point to ask you this question. What advice would you give to students that start their PhD and students that are about to leave their PhD?
So starting the PhD, okay, you have to choose a lab. The best way is that you have done a lab rotation or something like that in that lab that you are about to join. Then you can really judge how the atmosphere there is. So you have to find a lab where you like the atmosphere, where you like the people that you work with, and also it's very important that you like your colleagues and also your supervisor in order to communicate very often and very intensive with those people. I think this is a real important issue to be successful in the end, that there's a constant exchange, even on small things all the time. So things develop much better then. So that's important. As I said, if you know the lab before, you can make your own judgments. If you are about to join a lab that you don't know before, I would always advise invite one or two of the PhD students for a beer in the evening somewhere in a pub on a neutral ground, and let them talk and describe how they are doing in the lab. They're usually very, very honest about these things, and then you can make your decision. And then each supervisor has a different way of organizing the group, either directing it from above and having some hierarchical structures, or leaving the students more open or more freedom. But this also depends on the type of the students. Some like it to have a very rigidly planned experiment planned throughout the 3 years, where they know exactly what to do to finish their PhD. That's one thing. And then other students prefer to have a starting point, but then being allowed to develop the project in certain regions and applying methods that they think they would be suitable and things like this. So depending on the type and depending on the lab and the type of the project also, you have to somehow find a match between these things.
So this is the guys who are for the people who are about to start.
Yeah.
And what about for those that are about to leave?
I don't know. I mean, the typical thing is if you want to stay in academia, you need a postdoc. So one should probably, before handing in the PhD, trying to look around what could be interesting to follow as a topic. And then, I don't know, as early as possible, contact the potential PIs of the respective labs, because then they it sometimes needs some time to organize the money and things like this, and but otherwise, I mean, I think the same criteria would apply to join a lab for a postdoc than to join a lab for a PhD. You have to feel comfortable with the way the people work there and interact with each other.
And what about people who want to leave, for example?
Just apply for jobs.
Just apply for jobs.
Yeah,
contact people.
So at the moment, I think many companies are searching people, searching biologists or neurobiologists, being familiar with the typical lab techniques, and from my experience, the students that finished in my lab, master students mainly, they all found jobs in some pharmaceutical companies and stuff like this. But there are also, I think, also from my memories, there are lots of jobs that we are not aware that they are basically suitable for biologists, for example. So probably it would also be a good idea to other universities sometimes have this to invite people from different professions and kind of introduce what are the requirements to get into this and things like this. And you do this sometimes with the retreats, where you invite the alumni who took different career paths and kind of report their experiences, which is a good thing.
This was really indeed inspiring and also very informative to hear from people who are already successful in a different domain, and to find out, okay, what can be done in this field, in that field, am I suitable or not? And I found it very nice, and I hope that on the next retreat we have also some other people with other perspectives and learn about it. Okay, Professor Heinrich, we're almost at 1 hour and 30 minutes, and I think that we covered fairly a lot of topics. I really enjoyed this conversation with you, and thank you once again for being here and sparing your time with us and sharing with us all this nice and interesting information.
Yeah, thank you for this opportunity.
Yeah. See you then around soon, maybe at the retreat or maybe sooner, I don't know. Yeah.
I'm sure.
Thank you very much.
Thank you.
Neuroscience and Beyond. Know more, get inspired.